second part of the molecular basis of inheritance that started from the genetic code. During replication and transcription, a nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize. On the basis of complementarity, the process of translation requires transfers of genetic information from a polymer of nucleotides to a polymer of amino acids. Neither does any complementarity exist between nucleotides and amino acids, nor could any be drawn theoretically. There existed ample evidences how to support the notion that change in nucleic acids genetic material were responsible for change in amino acids in proteins. These led to the propositions of a genetic code that could direct the sequence of amino acids during synthesis of proteins. If determining the biochemical nature of genetic material and the structure of DNA was very exciting, the propositions and deciphering of the genetic code were most challenging. In a very true sense, it required involvement of the scientists from several disciplines: physicist, organic chemist, biochemist, and genetist. It was George Gamow, a physicist, who argued that since there are only 4 bases, and if we have to code for 20 amino acids, the code should be constitute a combination of bases. He suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. These was very bold propositions because a permutation combination of 4 into 4 into 4 would generate 64 codons, generating many more codons than required. Providing proof that the codon was a triplet was a more daunting task. The chemical method developed by Hargobind Khurana was instrumental in synthesizing RNA molecules with defined combinations of bases, homopolymers, and copolymers. Marshall Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. Severo enzyme polynucleotide phosphorylase was also helpful in polymerizing RNA with defined sequences in a template-independent manner. Enzymatic synthesis of RNA. Finally, a checkerboard for genetic code was prepared, which is given. The salient features of genetic code are as follows. The codon is triplet. 61 codons code for amino acids and 3 codons do not code for any amino acids, hence the function as stop codon. One codon codes for only one amino acids, hence it is unambiguous and specific. Some amino acids are coded by more than one codon, hence the code was degenerate. The codon is read in mRNA in a contiguous fashion. There are not pertinacious. The code is nearly universal. For example, from the bacteria to human, U would code for phenylalanine. Some exceptions to these rules have been found in mitochondrial codons and in some protozoans. AUG has dual functions. It codes for methionine and it also acts as inhibitor codon. Now try to opposite. Following is the sequences of amino acids coded by mRNA predict the sequence of nucleotide in the RNA. Do you face any difficulty in predicting the opposite? Can you now correlate which two properties of genetic code you have learned? Mutations and genetic code. The relationship between genes and DNA are best understood by mutation studies. You have studied about mutation and its effect in chapter 5. Effects of large deletions and rearrangement in a segment of DNA are easy to comprehend. It may result in loss or gain of a gene and so the function. The effect of point mutations will be explained here. A classical example of point mutation is a change of single base pair in the gene for beta globin chain that results in the change of amino acids residue glutamate to valine. It results into a disease conditions called as sickle cell anemia. Effect of point mutations that inserts or deletes a base on a structural gene can be better understood by following simple examples. Consider a statement that is made up of the following words, each having 3 letters that genetic code. Ram has red cape. If we insert a letter B in between has and red and rearrange the statement, it would be read as follow: Ram has breed cape. Similarly, if we now insert 2 letters at the same place by BI, now it would be Ram has beer ad app. Now we insert 3 letters together, say big, the statement would read. The same exercise can be repeated by deleting the letters R, E and D one by one and rearranging the statement to make a triplet word. The conclusion From the above exercise is very obvious. Insertion or deletion of one or two bases changes the reading frame from the point of insertion or deletion of three or its multiple bases. Insert or delete one or multiple codon, hence one or multiple amino acids, and reading frame remains unaltered. From that point onwards, such mutations are referred to as the frame shift insertion or deletion mutations. This forms the genetic basis of proof that codon is a triplet and it is read in a contiguous manner. TRNA, the adapter molecule. From the very beginning of the propositions of code, it was clear to Francis Crick that there has to be a mechanism to read the code and also to link it to the amino acids, because amino acids have no structural specialties to read the code uniquely. He postulated the presence of an adapter molecule that would on one hand read the code and the other hand would bind to a specific amino acid. The tRNA, then called sRNA soluble RNA, was known before the genetic code was postulated. However, its role as an adapter molecule was assigned much later. tRNA has an anti-codon loop that has bases complementary to the code, and it also has an amino acid acceptor end to which it binds to amino acids. tRNAs are specific for each amino acids. For initiation, there is another specific tRNA that is referred to as the initiator tRNA. There are no tRNAs for stop codons. In figure, the secondary structure of tRNA has been depicted that looks like a clover leaf. In actual structure, the tRNA is a compact molecule which looks like inverted L. Translation refers to the process of polymerization of amino acids to form a polypeptide. The order of sequence of amino acids are defined by the sequence of bases in the mRNA. The amino acids are joined by a bond which is known as a peptide bond. Formation of a peptide bond requires energy. Therefore, in the first phase itself, amino acids are activated in the presence of ATP and linked to their cognate tRNA, a process commonly called as charging of tRNA or aminosylation of tRNA to be more specific. If two such charged tRNAs are brought close enough, the formation of peptide bond between them would be favored energetically. The presence of a catalyst would enhance the rate of peptide bond formation. The cellular factory responsible for synthesizing proteins is the ribosome. The ribosome consists of a structural RNAs and about 80 different proteins. In its inactive state, it exists as two subunits, a large subunit and a small subunit. When the small subunit encounters an mRNA, the process of translation of the mRNA to protein begins. There are two sites in the large subunit for subsequent amino acids to bind to and thus be close enough to each other for the formation of peptide bond. The ribosome also acts as a catalyst. 23S-RNA in bacteria is the enzyme ribozyme for the formation of peptide bond. A translation unit in the mRNA is the sequence of RNA that is flanked by the start codon AUG and the stop codon encodes for the polypeptide in mRNA. Has also some additional sequences that are not translated and are referred as untranslated regions UTR. The UTRs are present at both fifth end before start codon and at the third end after stop codon. They are required for efficient translation process. For initiation, the ribosome binds to the mRNA at the start codon AUG that is recognized only by the initiator tRNA. The ribosome proceeds to the elongation phase of protein synthesis. During these stages, complexes composed of an amino acid linked to tRNA sequentially bind to the appropriate codon in mRNA by forming complementary base pairs with the tRNA anticodon. The ribosomes moves from codon to codon along the mRNA. Amino acids are added by one, translated into polypeptide sequences dictated by DNA and represented by mRNA. At the end, a release factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. Regulation and gene expression. Regulation of gene expression refers to very broad term that may occur at various levels. Considering that gene expression results in the formation of a polypeptide, it can be regulated at several levels. In eukaryotes, the regulation could be exerted at transcriptional level, formation of primary transcript, processing level, regulation of splicing, transport of mRNA from nucleus to the cytoplasm, translational level. The genes in a cell are expressed to perform a particular functions or a set of functions. For example, if an enzyme called beta-galactosidase is synthesized by E. coli, It is used to catalyze the hydrolysis of a disaccharide lactose into galactose and glucose. The bacteria use them as a source of energy. Hence, if the bacteria do not have lactose around them to be utilized for energy source, they would no longer require the synthesis of the enzyme beta galactosidase. Therefore, in simple terms, it is the metabolic, physiological, or environmental conditions that regulate the expression of genes. The development and differentiations of embryo into adult organism are also a result of the coordinated regulations of the expression of several sets of genes. In prokaryotes, controls of the rate of transcriptional initiation is the predominant site for control of gene expression. In a transcription unit, the activity of RNA polymerase at the given promoter is in turn regulated by interaction with accessory proteins, which affect its ability to recognize start size. These regulatory proteins can act both positively activators and negatively repressors. The accessibility of promoter regions of prokaryotic DNA is in many cases regulated by the interactions of proteins with sequences termed operators. The operator's region is the adjacent to the promoter elements is most operons, and in most cases the sequence of the operator bind in a repressor protein. Each operon has its specific operator and specific repressor. For example, LAC operator is present only in the LAC operon and it interacts specifically with LAC repressor only. The LAC operon. The elucidation of the LAC operon was also a result of a close association between a geneticist Francis Jacob and biochemist Jack Monod. They were the first to elucidate a transcriptionally regulated system. In LAC operon, here LAC refers to the lactose, a polycistronic structural gene is regulated by a common promoter and regulatory genes. Such arrangements is very common in bacteria and is referred to as the operon. To name few such examples: LAC operon, trap operon, arrow operon, his operon, well operon, etc. The LAC operon consists of one regulatory gene, the I gene. Here the term I does not refer the inducer, rather it is derived from the word inhibitor. And three structural genes Z, Y, and A. The I gene codes for the repressor of the LAC operon. The Z gene codes for the beta galactosidase, which is primarily responsible for the hydrolysis of the disaccharide lactose into its monomeric units, galactose and glucose. The Y gene codes for permease, which increases permeability of the cell to beta galactosidase, a gene enclosed a transacetylase. Hence, all the three genes products in the LAC operon are required for metabolism of lactose. In most other operons as well, the genes present in the operon are needed together to function in the same or related metabolic pathway. Lactose is the substrate for the enzyme beta galactosidase and it regulates switching on and off of the operon. Hence, it is termed as inducer. In the absence of a preferred carbon source such as glucose, if lactose is provided into the growth medium of the bacteria, the lactose is transported into the cells through the actions of permease. Remember, a very low level of expression of LAC operon has to be present in the cell all time, otherwise lactose cannot enter the cells. The lactose then induces the operon is the following manner. The repressor of the operon is synthesized all the time constitutively from the I gene. The repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing the operon. In the presence of an inducer such as lactose or alactose, the repressor is activated by interaction with the inducer. This allows RNA polymerase access to the promoter and transcription proceeds. Essentially, regulation of LAC operon can also be visualized as regulations of enzyme synthesized by its substrate. Regulation of LAC operon by repressor is referred to as negative regulations. LAC operon is under control of positive regulations as well, but it is beyond the scope of the discussion at this level. Now, Human Genome Project. In the preceding sections, you have learned that it is the sequence of bases in DNA that determines the genetic information of a given organism. In other words, genetic makeup of an organism or an individual lies in the DNA sequences. If the individuals differs, then their DNA sequences should also be different, at least at some places. These assumptions led to the quest of finding out the complete DNA sequence of human genome. With the establishment of genetic engineering techniques where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining DNA sequences, a very ambitious project of sequencing human genome was launched in the year 1990. Human Genome Project was called a mega project. You can imagine the magnitude and the requirements for the project if we simply define the aims of the project as follow. Human genome is said to have approximately 3 into 10 raised to the power 9 base pairs, and if the cost of sequencing required as $3 per base pair, the estimated cost in the beginning, the total estimated cost of the project would be approximately $9 billion. Further, if the obtained sequences were to be stored in type forms in books, and if each page of the book contained 1000 letters and each book contained 1000 pages, then 3300 such books would be required to store the information of DNA sequence from a single human cell. The enormous amount of data storage and retrieval and analyzing human genome project was closely associated with the rapid development of a new area in biology called as the bioinformatics. Goals of human genome project. Some of the important goals of human genome project were as follows: identify all the approximately 20,000 to 25,000 genes in human DNA, determine the sequences of the 3 billion chemical base pairs that make up human DNA, store these information in databases, improve tools for data analysis, transfer related technologies to other sectors such as industries, address the ethical, legal and social issues that may arise from the project. The Human Genome Project was a 13-year project coordinated by the US Department of Energy and the National Institute of Health during the early years of the HGP. The Wellcome Trust UK became a major partner. Additional contributions came from the Japan, France, Germany, China and others. The project was completed in 2003. Knowledge about the effects of DNA variations among individuals can lead to revolutionary new way to diagnose, treat and someday prevent the thousands of disorders that affect human beings. Besides providing clues to understanding human biology, learning about non-human organism DNA sequences can lead to an understanding of their natural capabilities that can be applied towards solving challenges in healthcare, agriculture, energy production, environmental remediation. Many non-human model organisms such as bacteria, yeast, Chinoraditis elegans, a free-living non-pathogenic nematode, Drosophila, the fruit fly, plants, rice, and Arabidopsis, etc. have also been sequenced. Methodologies. The methods involved to measure approaches. One approach focused on identifying all the genes that expressed as RNA, referred to as expressed sequencing text (EST). The other took the blind approach of simplifying sequencing the whole set of genome that contained all the coding and non-coding sequence and later assigning different regions in the sequence with function, a term referred to as the sequence annotation. For sequencing, the total DNA from a cell is isolated and converted into random fragments of relatively smaller sizes. Recall, DNA is a very long polymer and there are technical limitations in sequencing very long pieces of DNA and cloned in a suitable host using specialized vectors. The cloning resulted into amplification of each piece of DNA fragment so that it subsequently could be sequenced with ease. The commonly used host were bacteria and yeast, and the vectors were called as bacteria artificial chromosomes and yeast artificial chromosomes. The fragments were sequenced using automated DNA sequences that worked on the principle of a method developed by Frederick Sanger. Remember, Sanger is also credited for the developing method for determination of amino acids sequence in proteins. These sequences were then arranged based on some overlapping regions present in them. These required generations of overlapping fragments for sequencing. Alignment of these sequences was humanly not possible. Therefore, specialized computer-based programs were developed. These sequences were subsequently annotated and were assigned to each chromosomes. The sequence of chromosomes first was completed only in May 2006. This was the last of the 24 human chromosomes, 22 autosomes and XY to be sequenced. Another challenging task was assigning the genetic and physical maps on the genome. These were generated using information and polymorphism of restriction endonuclease recognition sites and some repetitive DNA sequences known as microsatellites. One of the applications of polymorphism in repetitive DNA sequences shall be explained in next section of DNA fingerprinting. Salient features of human genome. Some of the salient observations drawn from the Human Genome Project are as follows. The human genome contains 3,164.7 million nucleotide bases. The average gene consists of 3000 bases, but size vary greatly, with the largest non-human gene being dystrophin at 2.4 million bases. The total number of genes is estimated at the 30,000, much lower than previous estimated at 8,000 to 1,40,000 genes. Almost all 99.9% nucleotide bases are exactly the same in all people. The functions are unknown for over 50% of discovered genes. Less than 2% of the genome codes for the proteins. Repeated sequences make up very large portions of the human genome. Repetitive sequences are stretches of the DNA sequences that are repeated many times, sometimes 100 to the 1000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics and evolution. chromosomes. First has most genes 2968 and the Y has the fewest 231. Scientists have identified about 1.4 million locations where single base DNA differences SNPs single nucleotide polymorphism pronounced as SNPs occurs in humans. This information promises to revolutionize the process of finding chromosomal locations for disease associated sequence and tracking human history, applications, and future challenges. Deriving meaningful knowledge from the DNA sequences will define research through the coming decades, leading to the understanding of the biological system. These enormous tasks will require the expertise and creativity of tens of thousands of scientists from varied disciplines in both the public and private sectors worldwide. One of the greatest impacts of the having the HG sequence may well be enabling a radically new approach to biological research. In the past, researchers studied one or few genes at a time with whole genome sequences, and new throughput technologies we can approach questions systematically and on a much broader scale. We can study all the genes in a genome scale. Examples, the transcripts in a particular tissues or organ or tumor, or how tens of thousands of the genes and proteins work together in interconnected network to orchestrate the chemistry of life. Thank you.